Hey class, this is a review of karyotypes and meiosis. So this would be helpful for you if you missed some classes and you need to take the test. And I especially made this for 6th period because you're going to need review before you do the genetics test because we've been out of school for a while. Okay, so just a reminder, this is a karyotype. So remember this word karyotype. And this is a picture of homologous pairs of chromosomes. Okay, so each of these is a homologous pair. And remember this prefix homo means same. And these homologous pairs, you have one chromosome 12 from mom, and you have one chromosome 12 for dad. And you have that for all of your chromosomes. You have two sets of all of your chromosomes. So we say that our cells are diploid, di meaning two, because we have one chromosome from mom, one chromosome from dad. The same is we do have the same genes on these homologous pairs. However, the actual form of that gene could be different. The forms of genes, remember, are called alleles. And since mom and dad are not identical twins, for some genes, you have different alleles. So we would say that you're heterozygous for some genes, and other genes you're homozygous. This is another karyotype. In this case, see in this previous one, I can't go back. In this one, we have two X chromosomes and we don't have a Y, so this is a karyotype for a female. In the male karyotype, we had one X chromosome and we had one Y. The Y from dad, the X from mom. So for males, you have a Y chromosome which has very few genes on it. So actually the 23rd pair, these sex chromosomes for males, are not homologous. Most of the genes are here on the X, and you have a few genes on the Y, and that's what makes males males, but there's not as many. So you only have one copy of most of your genes. All of these other chromosomes are for your body, and we call these autosomes, meaning that they don't determine your gender, but your body. Okay, this is another karyotype, but here, look, the chromosomes look different. They look like X's. Okay, and these would be chromosomes in their duplicated state, meaning that what happened was DNA replication. You remember from the last unit when we would talk about prophase and mitosis, well, before prophase, that the chromosomes would line up and all this kind of stuff. So the DNA had to duplicate or make a copy, so we have a copy of that chromosome for the new cell. So you could see karyotypes where you have chromosomes in their duplicated state, and you could also see them in their unduplicated state. Those would be the other karyotypes that I showed you. So just to review, chromosome anatomy. This would be a chromosome in its duplicated state. Okay, and it's held together by the centromere. So this is that protein belt. Each of these arms we would call a chromatid. And since these are identical, we would call them sister chromatids. This whole thing is a chromosome. It is in its duplicated state. And if we were talking about a homologous pair, this would be, say, chromosome 12 also in its duplicated state. Okay, so this would be a homologous pair. This chromatid and this chromatid are not sister chromatids because they are not genetically identical. Okay, this would be a chromosome in its unduplicated state, and this would also be in its unduplicated state. So that's a homologous pair also, but those chromosomes are not in their duplicated state. Okay, so sometimes when chromosomes line up and they divide into the different cells, they don't divide evenly, and you can have more chromosomes than you should or fewer in some cases. Most of the time, this would result in development stopping for the baby. The baby it's not going to develop. For some reason, we don't know why, if you have an extra chromosome 21, we call that trisomy 21, the baby is born and does lead a relatively normal life, depending on the degree, and we call this Down syndrome. All right, many genes are affected, so remember, chromosomes contain 2,000 plus genes. So if you have an extra chromosome or if you're missing a chromosome, thousands of genes are affected. There's a whole suite of characters affected. Down syndrome, there's lots of things affected. The shape of the eyelids, the attachment of the tongue, the heart as well is a little bit different and unfortunately often has some problems. There's other types of chromosomal mutations. This is called a translocation. In this case, a piece of chromosome looks like 10 and chromosome 22 attach themselves to chromosome 9, maybe. I can't read the numbers here. And that causes a form of leukemia or blood cancer. So here they are kind of summarized. I gave you a summary sheet in class that had all of these. Deletion means a portion of that chromosome is deleted. Here, this portion is not present over here. A duplication, here we have B, this area of the chromosome is repeated. Here we have an inversion where the chromosome got twisted and now these regions switched. Translocation, so non-homologous chromosomes somehow switch their DNA and they are now in different places. That's a translocation. Okay, so this was the note sheet that I gave you in class. So pictures of homologous chromosomes, these are called karyotypes. What are homologous chromosomes? These are the pair from mom and dad, same genes, but they have different alleles. All right, forms of genes. Circle a homologous pair in its non-replicated state, so that would be there. Circle a homologous pair in its replicated state. This karyotype has them in its replicated state. They look like X's. Which karyotype shows a female? That would be this one. There's two X's. And the karyotype that shows a male is this one. You have an X and a Y. Circle all the autosomes in both pictures blue. Okay, so these would be all of the autosomes here. These are all of the autosomes here. And circle the sex chromosomes in yellow here and here. What does non-disjunction mean? So non-disjunction, we talked about that in class. You had to find an example. This is the uneven dividing of chromosomes. And generally, we talk about this during meiosis when we're making gametes. And that is the point of meiosis, to make gametes. All right, and what are gametes? What are these things? So these would be sperm, egg, the cells you need for sexual reproduction. But just think about other things too that may not be animals, so like pollen in plants, many types of spores and fungi and ferns and mosses and things like that. Okay, so those are gametes. This is a link to a video. Hopefully this works for you. I showed this in class. There's other videos on meiosis too. The Amoeba Sisters one is good. You can Google that one. But this is a much longer video, but from 6 minutes and 50 seconds to 10 minutes is the snippet that shows meiosis, and they explain it really well. Okay. All right. So this is the handout that I gave you in class, and if you remember, we used pink and blue. So I just have red here, but we used, remember, different colors to highlight the maternal and the paternal chromosomes, and we follow them throughout meiosis. And this sheet kind of goes like this. Ooh. So it's prophase, metaphase, anaphase, telophase, then metaphase, anaphase, telophase, and then we end up with 4 gametes, and I'm just going to make these look like sperm so you can remember that these are gametes. Okay. So just quickly, prophase 1, a big thing that happens that doesn't happen in mitosis is crossing over, and this is where the homologous pair kind of snuggles up next to each other, and they swap some DNA. Okay, now in the end, what's going to happen is you are going to, well, first you're going to do meiosis 1, and what's going to happen is this homologous pair, all these homologous pairs, you got 23 homologous pairs, they're basically going to split up. We called this chapter of meiosis the divorce, right? So they're going to split. And then in meiosis 2, the sister chromatids are going to split into separate cells. So what you're going to end up is you're going to end up with 4 gametes. You have all the genes. We're not missing any genes, but now you only have 1 copy of each gene. And that gene is either from the maternal chromosome or the paternal chromosome. And you multiply this by 23 because we have 23 pairs, and we have a whole lot of genetic diversity in our gametes, especially when you consider you have these crossed over bits in places. So this just shows you crossing over in a little bit more detail. And remember, this happens during prophase 1 of meiosis 1. So this is very much in the beginning of meiosis. And also in your notes, you should have a big, beautiful Venn diagram where you can compare meiosis and mitosis. So let's just go through that quickly. Mitosis, meiosis. You're going to have to keep these words straight, remember? And we talked about T. There's no T in meiosis, so think about T for 2 or twin, because we're making 2 cells. We're going from 1 diploid cell to 2 diploid. And they are genetically identical. It's just like a copying machine. However, meiosis, and meiosis has an E in it, which mitosis doesn't have, and we said E is 1/3 of the letters in sex. You're making 4 cells, and you're going from 1 diploid to 4 haploid, meaning you only have 1 copy of each chromosome, and you're making gametes. Here we're not making gametes, we're making body cells. Another word for body cells is somatic cells or somatic tissue. And they are not genetically the same. Not genetically identical. Okay. Most cells can do mitosis. All right, most cells do this, can replace themselves. However, the vast majority of cells in your body do not do meiosis. Meiosis is very specialized. It's only making these gametes. Okay, ovaries in females, testes in males. So only specialized cells do this. Only specialized cells do this. Let's see, some other things. What else do we want to say? We're doing PMAT, prophase, metaphase, anaphase, telophase just once. And here we're doing PMAT twice. These words mean, diploid means you have 2 copies of each chromosome, but we can write that down. And here we only have 1 copy of each chromosome. Stuff to put in the middle. We can say that DNA replication has to happen before this. And this happens in interphase. Oh, another thing is that crossing over only happens in meiosis. There is no crossing over. There's no lining up of homologous pairs in mitosis. So that's just a very quick summary. Hopefully you can find that in your notes because that's one thing we did in class. And this is just summarizing, like I was trying to say, this is meiosis 1, where you're dividing up the homologous pairs, and meiosis 2 is where you're separating those sister chromatids. Meiosis 2 is basically just like mitosis. All right, so that is a very quick summary for you.